What are proteins? They are a natural polymer that is the most versatile by far. They can be recognized in animal and plant products, such as meat, dairy, nuts and legumes, and eggs, which we will get into more detail later. Polymers. We can think of polymers as an intricate building made by bricks representing monomers, which are simpler molecules that combine to make these structures.
Monomers of proteins are called amino acids. They have a carbon center attached to a carboxyl group, an amino group, and another group or chain of atoms represented by R. This side group is very important, as it will establish the protein's shape and function and is unique to each type of protein.
These amino acids are linked together by peptide bonds, which connect the amino group of one monomer to the carboxyl group of another monomer. This is done through condensation reaction. There are a total of 20 naturally occurring amino acids. In our example of polymerization, we'll be bonding lysine and alanine. When bonding, the carboxyl will be on the left side of the peptide bond, and the amino group is on the right. This is common when joining any number of amino acids. Notice how the R group is different in each monomer. In this condensation reaction, we'll be focusing on these two groups. The byproduct of this reaction will form from the hydroxide atom of the carboxyl group and the hydrogen atom of the amino group, forming water. We are left with a peptide bond between the carbon of the carboxyl group and the nitrogen of the amino group. The double oxygen bond and the hydrogen bond will remain. But what are the conditions that cause this reaction? Because proteins are natural polymers, the molecular mechanisms within a cell create the conditions possible for peptide bonds to form between amino acids. Genes have instructions that control which specific amino acids join to form long polypeptide chains, which creates proteins' primary shape. Let's take a deeper look into the egg. Intermolecular forces create the structural properties of the egg's proteins. As said before, the primary structure is the chain of amino acids. Folding of these chains result in the secondary structure. Hydrogen bonds between amino and carboxyl group stabilize the structure. Further folding creates a 3D structure, also held together by hydrogen bonds. Another force present is the van der Waals forces. Van der Waals forces are weak electric forces that cause attraction between the neutral molecules. Focusing to the egg whites specifically, egg whites are composed of globular proteins whose 3D structures is like a sphere-like shape. Egg whites have both polar and nonpolar amino acids. The polar monomers are hydrophilic and love water. The nonpolar monomers are hydrophobic and are water-fearing. Because the proteins within the egg white cells are surrounded by water, the hydrophilic monomers are arranged on the outer portion of the globular proteins. So, how does the structure of the proteins impact the food? When these proteins are aggravated, they can become denatured, or in other terms, inactive. We can experience this while cooking. Heating or beating the egg whites causes the protein structures within to uncurl, as the energy inputted shakes the bonds apart. The way these bonds are broken allow you to cook and consume these proteins. This allows your body to obtain the amino acids it cannot produce.
Make sure to like and subscribe for more excellent content.